Hello, this is Holly Sanders, and welcome to this presentation on cells. This is for the ALHS 1011 Anatomy and Physiology course. This presentation correlates with Chapter 3 from your Merritt's Essentials of Anatomy and Physiology textbook. So at this point, you have already studied the introduction and you have taken a brief look at chemistry. So now, just like in the levels of structural organization, it is time to look at cells. And just a reminder, cells are what we're made of, and they carry out all chemical activities needed for us to sustain life. They are the building blocks of all things, and a group of cells will come together and create tissues. So just to give you an orientation of this next unit, we are going to cover cells and then move into tissues. The first thing we need to look at is the anatomy of a cell. Although they do not look the same, we're going to use this general picture of a cell or diagram of a cell that you see here in your lower right hand corner to describe all the anatomical parts of a cell. And then we'll look at some different cells and look at the differences in their shape and their anatomy in order to perform their specific physiological function. Well, the first notes that you need to take are the three main parts of a cell. The first part is the nucleus, and that is shown in purple here. The next is the plasma membrane, which is basically the skin of each cell around the outside. And then in the inside, there is what we call cytoplasm, which holds the different organelles of the cell. So these are the three main headings for the anatomy we're going to look at, starting with the nucleus. The nucleus is basically the control center of the cell, or what I like to refer to as the brain of the cell. It contains the DNA, and the nucleus also is comprised of three regions. Here's a large picture of the nucleus separated from the rest of the cell, and in this picture you can see the three main portions of the nucleus. Starting with the outside, we call this the nuclear envelope or nuclear membrane, and you can see it would be similar to the skin of the nucleus, keeping what is inside the nucleus separate from the rest of the cell. Then this purple part in the middle is what we call the chromatin, and this is the portion that holds the chromosomes. And then here in the middle is what we call the nucleolus, and this would be the site of ribosome assembly, and we'll talk further about what ribosomes are as we go through this presentation. So the three main portions of the nucleus again, the outside is the nuclear envelope, and you can see that there's little pores along the nuclear envelope, and this is actually where the ribosomes, which are created by the nucleolus, will come out, and again we'll talk about that further. And then the genes and chromosomes are held here in the chromatin. The second major anatomical portion of the cell is called the plasma membrane. And again, this is basically the skin of the cell that's going to keep the cell contents within it and keep it separate from its outside, very similar to our integumentary system. But as you recall from our introductory unit, we are made up of cells, so each cell has its own plasma membrane. There's a really fascinating portion of this membrane that helps control substances that can come into and out of the cell. And basically this is because the membrane is made of a double phospholipid layer. Now don't let this word be too intimidating. Use your medical terminology skills and put a slash through that O, and you'll see phospho, and then you'll see lipid. And most of you by now, especially after going through the chemistry unit, realize that lipids are the same as fats, and phospho is just from phosphorus. So this layer is simply made of a two-layer of phosphorus and lipids. And let's look at a picture of this. Here we go. This might be disorienting at first, but if you look, you see the little round heads here and little tails, and on the other side or inferiorly, there is another head and another set of tails. This would be the double layer. See, here's one and all the way through, and then below it, the second layer. And this allows our cells to regulate what goes in and out of them. For instance, if you're looking outside of the cell, here's the extracellular fluid, and there's a lot of water outside of our cells, as well as different electrolytes and substances that we need to control when they come into the cell. For instance, if you imagined all the water on the outside of the cell rushing to the inside of the cell, then the cell would explode. Or vice versa, if all of the fluid inside of the cell rushed to the outside, then the cell would shrink. So this needs to be heavily regulated. And the way this happens, if you look at this double phospholipid layer, see the heads, they are hydrophobic hydrophilic. Philic means loving, hydro means water, and you see the O, so it's a medical term. So basically they're pertaining to items that are water loving. Whereas the tails are hydro, and there's the O again, hydrophobic, which means water hating. So let's look back at the diagram. You can see how there's fluid on the outside of the cell touching the hydrophilic heads, which is just fine because they're water loving, so they're fine with touching this watery substance. And because it's a double phospholipid bilayer, there are heads again on the inside of the cell touching the fluid on the inside. However, these hydrophobic tails are essential because they're not going to let fluids just escape in or out of the cell. So the hydrophilic heads allow the substance to touch them, where the hydrophobic tails prevent any fluid from moving in between the plasma membrane. Now there's going to be a whole nother session where we look at how things do move in and out of the cell, but as for right now, I want you to be comfortable with the fact that the plasma membrane is made of a double phospholipid layer, which consists of hydrophobic tails and hydrophilic heads, and that they work together to keep things inside or outside of the cell. On the plasma membrane, there are some very specific projections that help to increase the surface area for absorption. Now understand that this might seem like a confusing term, but it's finger-like projections, and here is a picture of them under a microscope, and they're actually called microvilli, and they're sticking off of the plasma membrane, which is shown here. And what they do is increase the surface area for absorption. Well, basically what that means is on cells that contain microvilli, when they allow for more substances to go from outside of the cell, inside the cell, and through the plasma membrane. And we'll look at this a little closer. I just want you to be familiar with this anatomical structure. The next thing we're going to look at is how cells communicate with each other. And please remember that they have to communicate with each other in order to work together as a tissue. And remember, a tissue is multiple cells working under a common function. So if we look at this picture, I want you to look at what we've already learned. First of all, if you locate the nucleus, which is here, you can see the nuclear envelope, and you can see the little pores that the ribosomes will come out. And then you can trace the plasma membrane, starting up superiorly with the microvilli. You can see these finger-like projections sticking off the top. And again, microvilli's function is to allow a larger surface area for absorption of substance from the outside to the inside of the cell. And then if you follow this plasma membrane, you can see that these two cells are separated. Here it is, coming down, and finally around to the bottom, and this one coming up and to the side of the start of this new cell. And you can tell these are different cells because you can see the two nuclei and the plasma membrane around each one of them. But as you can see, these cells are connected, and they're going to function as a tissue, and they need to be able to communicate. Well, there's three areas, and I'm going to move back a slide, where the membrane or the plasma membrane has junctions, or where it connects. The first we're going to look at is called a tight junction. And this is an impermeable junction, and impermeable means that fluids cannot get through it. And basically it binds the cells together into leak-proof sheets. So if you imagine this being the mortar of bricks, then the mortar would be the tight junction where everything is held together. You can see that this comprises most of the plasma membrane. But it is really important that these cells do communicate so the tissue can work together and function together. And this is where this area here, called a gap junction, allows this communication. And I find this kind of easy to remember because if you look at these red dots, it kind of looks like an old telephone, like an old wall phone, where the receiver would have the dots in the earpiece and the mouthpiece. So this makes me think of the communication. Whatever information needs to be sent from this cell is going to go through the gap junction to this cell and through the gap junction to this cell and so forth. While again, the tight junction is just holding them together. And then finally, there's this really interesting junction called a desmosome, and it's an anchoring junction. So I want you to think of an accordion and how if you pull the accordion apart, it would be the desmosomes that allow it to stretch and then pull it back together. So if you think about the different cells of our body, consider how few desmosomes bone cells would have compared to how many skin cells would have. You can pull and stretch your skin or your muscle where bones hold tight. So depending on the function of the cell, they're going to have a differentiating number of desmosomes. So again, the three membrane junctions that hold cells together would be the tight junction, the desmosomes, and the gap junctions. So far, we've covered the nucleus, which is comprised of the nucleolus, the chromatin, and the nuclear envelope. And then we took a quick look at the plasma membrane, including its junctions, and the microvilli, which stick off the plasma membrane to help with absorption. And we talked about how it's made of a double phospholipid bilayer that allows substances to be regulated when they come in and out of the cell. Our third main anatomical component of the cell is called the cytoplasm. And basically, the cytoplasm is everything in between the nucleus and the plasma membrane. So it's all of this in between. The cytoplasm also has three major elements within it. The cytosol, and that would be the jelly type of substance that's holding everything up. The organelles, and that would be the little organs. If you break this word down, L's means little, so little organs within the cell. And then inclusions, which are basically chemical substances such as stored nutrients or cell products. And in the purposes of this course, we're not going to really get into inclusions, but I want you to be aware that they are within the cytoplasm. So let's take a moment and look at the cytosol and organelles. Here's the basic picture of a cell that you're going to be working with. This is the diagram you're going to be filling out, and you'll see again on your test. And remember that not all cells are set up the same way, but there are similar anatomical components within each one. In review, here's the nucleus and its three parts: nuclear envelope, chromatin, and nucleolus. And on the outside is what we call the plasma membrane. Here's the microvilli on the plasma membrane. And as I said before, everything between the nucleus and the plasma membrane is the cytoplasm. So the cytoplasm holds all of the organelles. And here is an organelle, here, here, here. And then if you see this kind of yellowy orange fluid, that would be the cytosol. And that's what's holding up all the organelles. Because if you think about gravity, if there wasn't something suspending them, then they would just fall to the bottom of the cell and they wouldn't be able to do their function. So now let's take a moment and look at the basic physiology of each of these organelles. First one we're going to start with is called the mitochondria, which is commonly called the powerhouse of the cell. The basic function of the mitochondria is where ATP is made. And ATP, as we studied in the last unit, is cellular energy. One of the memory techniques that I use for the mitochondria is that they actually look like little sausages to me. And if you think about eating, that would provide energy. So these little sausages help provide ATP, which is cellular energy. The next organelle we're going to look at are called ribosomes. And this is basically the site of protein synthesis. So let's recall what synthesis is from our chemistry unit. Synthesis is basically when you bring two things together to make something larger. So if you imagine having Legos and putting them all together, you'd be synthesizing those Legos. So in this case, protein synthesis is when amino acids, which are the building blocks of protein, are synthesized and brought together to make a larger protein. And as you know, protein creates the structure of our body, so this is very important. This is going on regularly within all the cells. There's two places ribosomes are found. The first is along this maze-like network, the blue here, which is called the endoplasmic reticulum, and here it is, inside the plasma reticulum, endoplasmic reticulum, and we actually call this rough endoplasmic reticulum because the ribosomes make it bumpy or rough. Again, the function of a ribosome is protein synthesis. Ribosomes, if you recall, are created within the nucleolus of the nucleus, and then they're sent out through the nuclear pores of the nuclear envelope, and they live along the rough ER, and this is where protein is synthesized. The other site you'll find ribosomes is just floating within the cytosol, as seen right here. This little gathering of red dots would be loose floating ribosomes. The next organelles we're going to look at are the endoplasmic reticulum. We've already discussed the rough endoplasmic reticulum. It holds the ribosomes, and it is a site for protein synthesis. But over here, this is called endoplasmic reticulum, but in this case, it's smooth because it doesn't have ribosomes. The function of smooth endoplasmic reticulum is lipid metabolism and the detoxification of drugs and pesticides. So rough ER, or endoplasmic reticulum, has ribosomes and it synthesizes proteins, whereas smooth ER functions in lipid metabolism and the detoxification of drugs. Next, we're going to look at the Golgi apparatus. And the Golgi's main job is to modify and package proteins. And let's take a look at this. The Golgi is the green substance here. And as you can see, it's closely connected to the rough ER, which is essential because, as we just learned, the rough ER is studded with ribosomes, and it is the area for protein synthesis. So once the proteins have been synthesized, they're going to come to the Golgi apparatus, and I like to consider the Golgi the UPS of the cell. As you would take a package to the UPS store, they package it up for you and ship it off to where it needs to go. So if you look at this area of the Golgi, you can see it is breaking off with substances inside of it to get to the outside of the cell to be released. And we're going to look at this again closer in a different presentation, but for the purposes of this presentation, please just realize that the Golgi apparatus is the packager of the cell. One of the packages that the Golgi apparatus makes is called a lysosome. Basically, a lysosome contains enzymes that digest worn out or non-usable materials in the cell. And just like our entire body creates waste, so does our cell. So here on this diagram is an example of a lysosome, and this has waste products within it. I consider the lysosome the garbage man of the cell because it's going to remove the waste from the inside of the cell. Another package is called a perioxisome, and basically that is a package that detoxifies harmful substances such as alcohol and formaldehyde. And the last couple of things we're going to look at are called vacuoles and vesicles. Vacuoles, they operate kind of as a storage freezer. My grandparents always had one. Go down in the basement, open up the freezer, there's a ton of frozen meats and foods in there that they're storing for a separate time. And this is what the vacuole does for the cell. Basically, it's a storage compartment within the cytoplasm for substances that aren't currently being used. And then a vesicle is something that carries contents in and out of the cell. And we're going to look at this in more detail in a different presentation, but basically this is the box or what the Golgi apparatus is packaging. Finally, let's take a look at what gives the cell its shape. The basic shape of each cell comes from its cytoskeleton, and you probably see the med term O right here. Cyto means skele, and skeleton is framework, so it's the basic framework of the cell. There are also certain cellular projections that are found on the outside of the cell, but not all cells, only the ones that need them to perform their function. One of these substances is cilia, and basically that moves materials across the cell surface. Something we're going to look at further as we go through the course is where our mucous membranes are. And basically they're anywhere that has an opening to the outside of the body. So the mouth, nose, ears, rectum, anywhere where skin doesn't cover is covered by a mucous membrane. And the cells that form the mucous membrane are covered with something called cilia. And basically cilia moves substances across the surface of the cell to help keep them from getting inside. This would be something like a bacteria that we've breathed in, so we can then sneeze or cough it out, or that we can just keep it from getting inside of our cells. Another cellular projection is a flagella, and this is a tail that propels the cell, and only one cell has a flagella, and that is the sperm. So as you can see, sperm is a cell. Here is the nucleus and the flagellum. And as we talked about a minute ago, cells have different shapes according to their cytoskeleton, and they also have different organelles inside of them according to their function. So here's an example of a fibroblast, and this helps in our clotting functions. But if you look, you can see there's a nucleus, there's a rough ER, and here's the Golgi apparatus. And the cell to the right is actually a erythrocyte. Erythro means red, cyte means cell, so it's a red blood cell. And we'll look at this further when we get into the blood section, but the erythrocyte doesn't have any organelles, and we'll discuss this later. So just to show you, depending on the cell's function, they do have different shapes and different anatomical components. Here is a macrophage, which is one of our white blood cells, and you can see this macrophage has a lot of lysosomes. And if you remember, lysosomes are the garbage men of the cell and getting rid of waste. It would make sense that when a macrophage comes along and attacks bacteria, that it needs a way to package and get rid of it. So it has multiple lysosomes. Here is a nerve cell. And although this looks quite different than the cell we were originally studying, you can see again a nucleus and rough ER. So now it's your turn to learn these anatomical components of the cell. What I'm going to want you to do is get out your cell diagram, which I've provided for you, and label all these main parts that we covered. And that's the anatomy. The labeling, again, is just looking and defining the structure. And then I want you to also say the physiology or the basic function of the cell. Let's go back through, label the anatomy, and describe basic physiology. And just as a memory technique, don't give full definitions, just give the shortest amount you can use so you can retain it. Thank you for watching this presentation. Our next one will be on membrane transport.